Good afternoon, ladies and gentlemen. My name is Rudi Glocksuber. I'm representing the Department of Biology as the head of this PhD exam. And I would like to welcome you to today's PhD defense of Mr. Devang for his PhD thesis in the group of Professor Wilhelm Grissom. Just a quick information for those of you who are not familiar with the procedures. We will have a 20-minute or so presentation of the candidate on his most important results. And after that, there will be a public discussion where anyone, except the examiners, everyone is invited to ask questions. And you are all encouraged to do so. It will not have any influence on the outcome. After that, I will ask the audience to leave the room, and then you will have to wait outside until the closed oral exam is finished, which will take at least 40 minutes. So with this, I think we can start, and I would like to invite you now to present your most important results. Thank you.
Okay, welcome everyone. I'm going to be talking about my PhD thesis, which is titled Assessing Genetically Engineered Resistance to Cassava Gemini viruses using virus population sequencing. My project was divided into three main parts. In the first part, I studied the performance of antiviral RNAi technology, so this is existing technology, and I saw how the technology performed in field conditions. In the second project, I tried to create a new mechanism for conferring immunity by repurposing a bacterial immune system in a eukaryotic plant. And then in the third project, I tried to improve existing RNAi technology using DNA replicons to give enhanced resistance to cassava Gemini viruses. For this talk, I'll be talking only about the first two projects, and I'll start with the field work. So our lab works on cassava. Cassava is an important tropical staple food crop. It's grown all across the tropics, and it originates in South America. However, right now, the major production is actually in Africa, so more than 50% of cassava production worldwide is in Africa. The crop is grown mainly for its starchy tuberous roots. These are eaten both by humans, but also used for animal feed, as well as industrial starch. Production of cassava is severely restricted by a disease called cassava mosaic disease. This disease is mainly located on the African continent, but also in India and Sri Lanka. However, there are reports which suggest that it could potentially spread to other cassava growing regions across the world. And in fact, only in 2016 that there was a first report of this disease in Cambodia, so more east than it has been reported previously. The disease is spread by whiteflies. So these are these tiny insects that feed on the cassava leaves. They transmit the virus directly into the phloem stream of the leaf, and then the disease causes severe symptoms and then average yield losses of 15 to 24% in Africa. The virus itself is comprised of two genomes, a DNA A genome and a DNA B genome. Both the genomes are single-stranded DNA molecules, and each codes for separate complement of genes which are required for the virus to replicate as well as to move from cell to cell in the plant. The virus is actually a complex of different virus isolates and species. So far, 9 species of this virus have been reported in Africa, and they're divided roughly into 3 clades, which is in blue over here, the African cassava mosaic viruses, which were originally identified in West Africa, the East African cassava mosaic viruses, which were identified first in East Africa and Tanzania and Kenya, and then the South African cassava mosaic viruses, which are mainly found in South Africa, Madagascar, and Mozambique. So in 2009, our lab produced the first transgenic plants that were resistant to this virus. In this study, the scientists from our lab used a technology called RNA silencing or RNA interference. The way this works is they introduce a transgene into the plant, which expresses double-stranded RNA molecules, long double-stranded RNA molecules, which are then plant then processes into small, short RNAs, which are either 21 to 22 nucleotide long fragments or 24 nucleotide long fragments. These then either cause the degradation of the virus's mRNA, hence preventing the virus from producing protein, or they cause directly DNA methylation of the virus's genomes, preventing it from replicating as well as transcribing its genes. These were the results from 2009, and as you can see, the transgenic plant looks much more healthier compared to the infected control plant, where you can see the virus symptoms of this disease. These same plants were then reassessed over the years. So this is a report from 2014. It's unpublished data from another postdoc in the lab. He monitored the infection of transgenic plants, which are shown at the bottom, as well as the control plants, which are these two lines up top. And he monitored how the percentage of plants that were infected over a 9 weeks period. And as you can see, the transgenic lines perform much better than the control plants. So the transgenic lines range between 40 to 30% of the plants were infected, whereas for the control lines we had almost 100% infection. So at least in the glasshouse, under infection by a single virus isolate, we were able to observe that these transgenics are still resistant to the virus. So the next step was to establish a field trial of the virus, of the virus-resistant plants in Africa itself. And my supervisors did this in 2013-14. So there was this confined field trial that was established in Kenya, right at the border of Kenya and Uganda, in collaboration with scientists from the Masinde Muliro University of Technology, which is located right at the field site. I analyzed the results of this trial. These are the results in terms of, so on the top panel, I have the percentage of plants that were infected for control lines as well as transgenic lines over a 30-week period. The second panel shows the same data, but in terms of the average symptom severity per plant line. So as you can see, the control plants did show infection early on in the experiment, whereas the transgenic plants did not show symptoms until week 15 or 17. At the last time point of the field trial, the transgenic plants did not show significantly better results compared to the control plants. It's important to note that during the trial, as shown as this blue bar over here, there was a severe hailstorm as well as a flooding event, which might have contributed to the infection after this time point. Also, at the same time point, we observed a doubling in the number of whiteflies that were present on all the lines in the field. When we got these results, our first hypothesis was that the reason these transgenic plants failed in the field was that there might have been multiple geminiviruses, species or isolates, which then together contributed to loss of resistance. So in order to validate this hypothesis, the first experiment I performed was to try and sequence what viruses were actually present in the transgenic plant lines as well as the control plant lines. So when we consider the problem of sequencing virus populations in a plant, you have to remember that a plant cell might be infected with either a single virus isolate, but more often with multiple virus isolates, some of them differing by a small amount and some of them differing by a large amount. Plus, when you extract DNA from infected material, you have a lot of chromosomal DNA as well. And by my calculations, we have about 2,000 to 5,000 times more chromosomal DNA compared to wild DNA, depending on the amount of virus infection. So there are two steps required to then sequence these viruses accurately. One is to then filter out the wild DNA from the background plant chromosomal DNA. So we need to enrich the wild DNA without any bias. And the second step is to then sequence these viruses that we have enriched accurately. Now, the most popular sequencing technology called Illumina sequencing produces very short sequencing reads. And then the challenge is to then computationally reconstruct such a population of viruses using this mixture of short reads. So this is practically impossible. And that's why the first step that we had to do to actually sequence the viruses was to develop a new sequencing method. So we developed a method which we call CytoSeq, which stands for Circular DNA Enrichment Sequencing. The method has both The method has both a new wet lab protocol for enriching virus molecules, virus DNA without any bias, as well as and then we sequence the viruses using a next generation sequencing technology called PacBio, which produces long sequencing reads unlike older technologies. And then these sequences need to be processed by a custom algorithm which we designed, which then allows us to pass these long reads and then return back to the circular DNA sequences. So I won't go into the details about how the protocol works, but our enrichment results look very promising. So as you can see, in 3 separate runs, we are able to enrich viral DNA by more than 98%. And then when we look exactly at what sequences were present in infected transgenic plants as well as control plants, these are what the results look like. So in this graph, I'm showing you the distribution of different viruses in each plant line. So each horizontal bar is a single plant. At the bottom, you have 3 control plants, and at the top, you have transgenic plants. What we immediately saw was that in the control plants, we had 2 populations of viruses. We had a major population over here, which is more similar to the virus that we use in the lab for infections. And then we had a second population of viruses, which is only 70 to 75% similar to the virus we use in our lab. And what we could see was that this population is clearly eliminated in the transgenic plant lines, so the transgenics do work against this population of viruses. But this leaves space for this second population, which then occupies the full, which is then present at 100% of the virus in this plant, in the control plant, transgenic plant lines. So what we could conclude was that the RNAi does work, but it works very well only against the target species or viruses that are very closely related to the target species. Whereas the second species of virus that we detected in the field is left uninhibited in the plants. We then also performed a phylogenetic analysis of what viruses were present in these samples. And what we were able to determine was that the 2 populations in the control plants consisted of a population that was related to the African cassava mosaic virus, which is mainly from West Africa, as well as the East African cassava mosaic virus in green, which is mainly found in East Africa. However, in the transgenic plants, this first population of viruses that are closely related to the African cassava mosaic virus species is completely eliminated, leaving behind only the East African type viruses. So in summary, for this project, we developed a sequencing technology that could give us full-length viral genomes in one sequencing read, which means we do not have to do any computational assembly of short reads to reconstruct virus genomes. This means that we are able to sequence 1,200 virus genomes, full virus genomes, which is twice all previous data in the field. More importantly, we are able to reduce the cost per genome from $40 to $1.5, which is 16-fold reduction in cost, which then allows us to get this done. Importantly, the way we design CytoSeq, it can work for any circular DNA, so not just the viruses that we work with, but also other circular DNA viruses, as well as extra-chromosomal circular DNA like plasmids, etc. For the RNAi field trial, we use CytoSeq to show that RNAi technology is indeed very effective, but only against viruses that are related to the target species. So at the same time, I was carrying on with the second project, which was to develop a new technology for virus resistance by repurposing a bacterial immune system for this purpose. So the immune system that I was working with in bacteria is called CRISPR-Cas9. The way this system works is you have a Cas9 protein, which is shown in yellow over here in the background. And the protein then complexes with a second RNA molecule, which is the guide RNA in blue over there. And the protein and guide RNA complex then target a complementary DNA sequence on the plant genome, or any DNA sequence, in fact, and then cleave this DNA molecule. So prokaryotes like bacteria use this very often to defend against invading viruses as well as plasmids. Because once a virus enters a bacterial cell, the system can then cleave the virus's DNA, preventing the virus from replicating. In 2013, this tool was then re-engineered for use in eukaryotes, the purpose of which was to then use this to make targeted cuts on the eukaryotic genome to basically edit genomes of animals as well as plants. This video shows very well how the system works. So you have the guide RNA complex binding to the DNA molecule, and then once it's active, you can see it cleaves the DNA so that you have two pieces left over. So our idea was to try and use this as an immune system in eukaryotic species. So in our case, it was cassava. The way our system works is we would introduce a transgene which would express both the Cas9 protein as well as the single guide RNA, which is shown over there. These two then would form a complex within the plant, enter the nucleus, and then cut the replicating DNA of the virus. Now, we did not know at the time that if the virus DNA was cleaved, whether it would get repaired, what proportion of it would get repaired, or would all the cleaved fraction just be degraded. And this left us with the second question, which was, could the system work efficiently enough to prevent the virus's DNA from replicating further to cause disease? So in order to do this, we designed several guide RNAs to target different areas of the virus genome. So these are different genes on the virus genome, and you can see we designed guide RNAs that could cleave almost all of these genes. It was very crucial that we designed guide RNA that could not cleave the cassava genome, but could only cleave the virus genome. We then looked at the efficiency of this guide RNA using an in silico program, as well as we tested it in vitro in the lab. So this is the in vitro cleavage assay results. And what you can see here is the top band is the intact virus DNA. And then when we introduce the Cas9 plus either one of these guide RNAs, it cleaves the virus to release these two second cleavage products of the virus. Based on these results, as well as based on the in silico assay, we decided to construct transgenic plants that would express the Cas9 protein as well as the first guide RNA over here, which is guide RNA1, because it was very effective in vitro as well as predicted to be very effective in silico. We then produced transgenic lines expressing these constructs. So we had a control construct which only expressed the Cas9 protein but no guide RNA, so it's a defective system. Then we had a test construct where we had the Cas9 protein as well as guide RNA1, which was expressed in the plant. So we developed 2 plant lines of cassava expressing the control construct, as well as 7 transgenic plants expressing the second construct, the test construct. We are able to show in this picture that we are able to express the full-length Cas9 protein in all of these lines, as well as using qRT-PCR, we are able to show that in the test lines we have expression of guide RNA1, which is obviously absent in the control lines. So then we took these transgenic plants to the greenhouse and infected them with the virus, with a single virus isolate in our lab. This panel shows you the progression of the infection in these transgenic plants over 8 weeks. On the left, I've shown you the infection rate every week for individual plant lines belonging to the test or control batch. What you can see is that we do have some lines, like line 139, that do seem to show less infection rate compared to the control plant, but overall we do not see any significant difference in the proportion of infected plants between the test construct as well as the control construct. When we also look at the severity of the symptoms in each plant, we see the same result. We do not see any significant difference between the control plants as well as the test plants. So in order to investigate why the system was not effective or as effective as we expected, we deep sequenced the viruses that infect each of these plant lines, the control lines as well as the test lines. In total, we generated 5,000 genome sequences, and then we analyzed these sequences. So in this figure, each horizontal line is one virus genome, and wherever you see a peak, it means that there's a mismatch between the virus genome that we detect in a plant and our reference virus genome, which was used to infect. This means that in the test lines over here, these virus genome molecules, which have these peaks, were in fact edited by the CRISPR system, and then there was a repair of the virus giving us a mutation, which we can detect with our sequencing experiment. What you can see over there is that in our best performing line, in terms of editing, only 11% of the virus genomes were edited. So this is obviously less than optimal. Secondly, we then looked at what types of mutations were actually present in these plant lines due to the CRISPR system. Because, so we looked at the edited sequences in each plant line. So I'm not showing all the plant lines in this presentation. And then we analyzed the frequency of substitutions mutations in these plants. What we found was that the CRISPR-Cas9 system produced a new virus mutant in 3 independent plant lines, and it was the same mutant in all 3 plant lines. And it was present at a very high proportion. So in this line, 48% of all edited viruses contained this mutant. And we were also able to show that this mutant was in fact resistant to further cleavage by the Cas9 system. So to us, this means that the CRISPR actually induced the evolution of for a resistant virus strain in these plants. This mutation was not found in any of the viruses infecting the control plants. In conclusion, we developed CytoSeq, which is a cost-effective and accurate method for deep sequencing circular DNA. We then used this to analyze RNAi plants in the field, and we found that it's actually very effective against greater than 90% similar viruses, so viruses that are more than 90% similar to the target virus. But this leaves us with the problem, which is that we need to analyze virus diversity in the field before we design transgenic plants to take it into the field. For the CRISPR system, we had both inefficient cleavage of the CRISPR system against the virus, and we found that the CRISPR actually induces the evolution of a new cleavage-resistant virus strain. With that, I'd like to finish by thanking my supervisors, Willie and Hervé, my students, Alessandra and Mariam. Alessandra helped a lot with the Cas9 project, as well as Mariam was a Kenyan student who helped a lot with the field trial analysis. Also, my collaborators, Matthias, who's in our lab, and helped a lot with developing the software behind CytoSeq technology. Professor Hassan Ray, who is a collaborator on site in Kenya, and Andreas Patriani, who's from the Functional Genomics Center in Zurich, and helped implement CytoSeq technology. I'd also like to thank all the Cassava group members, as well as the whole Plant Biotech group. And I had partial funding from the European Union, so I'd like to acknowledge that. Thank you.
Thank you very much. Questions?
Perfect seminar.
So if there are... okay, good. Please.
Did you use your deep sequencing on the RNAi plants to see if you had the evolution of any mutations in the field as well? Or were all of the infecting viruses pre-existing variants?
So our CytoSeq technology gives us 99% accurate sequences. So we did see 1% divergence in the sequence results, but there was no mutation greater than 99% from the control plants.
Okay, so all of the viruses which were found in your RNAi plants were all naturally
occurring.
Why do you need this 90% for the cytosequencing mechanism?
That's not for the sequencing, but for the RNAi technology. And we think it's because RNAi technology is based on the complementarity of the hairpin that is produced in the plants, the double-stranded RNA. It has to be nearly perfectly identical to the virus's mRNA sequence. So we found, for the first time we were able to quantitate this, that we need more than 90% similarity to have the system actually working in the plants.
Yes?
I'm not sure I really get how many guides you use for the CRISPR approach, but what about
we tested
using several guides in the same construct?
Yes, so I tried to create constructs with several guides, but we were not able to generate transgenic plants. But it is possible that if we generated several guides, they could create more severe mutations, which would then perhaps limit the rate of evolution of new viruses. But because the cleavage efficiency itself is so low, I don't believe that that just having more guides would be enough to give us resistance, because we only detect up to 11% efficiency, and I don't think that's enough. Also, the virus can replicate through recombination as well. So it's also possible that if we cleaved out larger portions of virus, that they could still contribute to replication of other viruses instead of present.